So oxygen is transported in the blood, and it is transported via a molecule called hemoglobin. And hemoglobin is considered our respiratory pigment. And here we're talking about vertebrates. Some invertebrates have a different respiratory pigment, a different mineral, which actually gives their blood a different color, like blue or green. When we talk about this respiratory pigment, it is in vertebrates, it is transported in red blood cells. And these red blood cells are called erythrocytes. And these red blood cells are packed with hemoglobin. And in fact, in mammals, they're so full of hemoglobin that they actually lose their nucleus and are incapable of reproducing themselves. So they have to be produced continually by the red bone marrow in our bones. We can also talk about how this respiratory pigment carries oxygen. And so we can talk about the binding of oxygen to hemoglobin and how this is a reversible thing that happens here. So it's very important that hemoglobin can pick up oxygen, but it's also equally important that it can release it into the tissues where oxygen is needed. And so this releasing of the oxygen from the hemoglobin has a name, and it is called dissociation. And this is really important because we can talk about the conditions under which hemoglobin will dissociate from oxygen and give it up to the cells that need the oxygen. So we say that hemoglobin binds reversibly with oxygen. If we look at the molecule of hemoglobin, this is just one that I happened to take off the internet, you can see that it has protein structures. So the globin is protein, and it is globular protein. It has a very specific three-dimensional shape. And when we look at that, you'll notice that there's actually 4 different globins. They're actually labeled beta and alpha, beta 1 and alpha, beta 1, beta 2, alpha 1, alpha 2. Okay, so they have different characteristics. We also have the heme group. And this is important because this is actually where oxygen binds. And in vertebrates, it contains iron, which is in the periodic table Fe. So this shows the whole heme group right here. You'll notice that there's nitrogen and other organic elements attached to this. So it's not just iron. So when we talk about hemoglobin saturation, this is an interesting concept. And it also plays a role in determining whether hemoglobin is picking up oxygen or whether it is releasing oxygen. So if it is 100% saturated, that means that all of the heme groups, all of the iron groups have oxygen attached. So all 4 heme groups have O2 attached. And this is really important because you don't want your hemoglobin saturation to fall even below 90. And in times of COVID, you know that this is one of the characteristics of COVID, that people can have their saturation levels because they're not breathing properly. Their saturation levels fall really low really quickly, and that is why it is so deadly. And so there is a way to measure your hemoglobin saturation, and you might have had this done at the doctor's office. It can be done just with what is called a pulse oximeter, where you put it onto your finger, and it actually uses light to determine your hemoglobin saturation. And so if it is 50% saturated, then you're only carrying or oxygen is only bound to like half of the hemoglobin molecules, or half of the heme groups. So if we look at this saturation, this is what is in this diagram. So this is hemoglobin saturation. So if it is at 100%, we see that that is generally where your partial pressure of oxygen is very high. So this would like be in the lungs where you're picking up. That is where you're picking up oxygen. And then you'll notice that it has a weird shape. It's not like linear, but it goes very slowly in its dissociation. It gives it up very slowly, and then all of a sudden it gives it up really readily. Like right here, the slope of this curve becomes more dramatic, right? And so that is where we have the tissues. So this is like metabolically active tissues. So if we look at the slope, we can see, and in this diagram, it shows that if there is a decrease in partial pressure, if there is a decrease in pH, which actually has to do with levels of CO2, which we'll talk about in a minute, if there is an increase in temperature, all of these factors would cause hemoglobin to releases or to dissociate from oxygen. So you could ask, well, what does pH have to do with it? So when we look at blood pH, it has to do with CO2 levels in the blood. So when we look at blood pH, it is generally about 7.45 on the pH scale, which you'll remember from maybe taking a chemistry class, that is actually slightly basic or slightly alkaline. And so when carbon dioxide combines with water, it produces a weak acid, which is carbonic acid. So this is carbonic acid. This acid will actually dissociate into hydrogen ions plus bicarbonate ions. So this is a hydrogen ion, and that is what determines pH. The greater the hydrogen ion concentration, the lower the pH. It's inverse proportion. And this is my bicarbonate ion. So in metabolically active tissues where CO2 is being released into the circulatory system, we're going to get the production of carbonic acid, and then it's going to dissociate, and it's actually going to lower the blood pH. And we'll notice that in lower blood pH, it actually changes the shape of this curve. So here, they call this the Bohr Shift, where hemoglobin actually will behave differently under low pH, high temperature. So you'll notice here that under low pH, it actually releases its oxygen much more readily. And that is different when we look at low CO2, high pH, then it doesn't release it as readily. And so that is how hemoglobin and oxygen dissociate